Bioinfocongress II
Çalıştay. Doktor Bora Uyar. Kanser hasta stratifikasyonu için çoklu omik veri entegrasyonu. Doktor Bora Uyar, lisans eğitimini Sabancı Üniversitesi Biyolojik Bilimler ve Biyomühendislik Bölümü, yüksek lisans eğitimini Simon Fraser Üniversitesi Bioinformatik, doktora eğitimini Avrupa Moleküler Biyoloji Laboratuvarı Yapısal ve Hesaplamalı Biyoloji programında tamamlamıştır. 2015 yılında Max Delbrück Center Berlin Tıbbi Sistemler Biyolojisi Enstitüsü'nün Bioinformatik ve Omik Veri Bilimi platformunda göreve başlamıştır. Bioinformatik disiplinini kullanarak insan hastalıklarını moleküler düzeyde anlamaya çalışmak araştırmalarının esas amacıdır. Özellikle kanser biyolojisi üzerine hem toplu hem de tek hücre düzeyinde ölçülen multi-omik verileri entegre etmektedir. Doktor Uyar, şu anda tek hücreli dizileme veri kümelerini kullanarak hücresel kimliklerdeki bozulmaların nasıl çözüleceği ve bu bilginin TCGA ve ICGC, PCAWG gibi büyük ölçekli insan dizileme projelerinde bulunan toplu dizileme veri kümelerine nasıl aktaracağı ile ilgilenmektedir.
Bioinfocongress II
Workshop. Dr. Bora Uyar, Multi-Omics Data Integration for Cancer Patient Stratification. Dr. Bora Uyar is a bioinformatics scientist at the Bioinformatics and Omics Data Science platform of the Berlin Institute for Medical Systems Biology of the Max Delbrück Center in Berlin. He holds a bachelor's degree from Sabancı University, master's degree from Simon Fraser University, and a doctorate degree from European Molecular Biology Laboratory. His main aim is to apply his skills in bioinformatics to further our understanding of human diseases at the molecular level. He is currently interested in studying cancer biology and the immune system, applying multi-omic data analysis strategies at both bulk and single-cell level resolution.
Sayın Dr. Bora Uyar ve değerli katılımcılarımız, Bioinfocongress II Çalıştay etkinliğimize hepiniz hoş geldiniz. Değerli hocamız bugün bizlere Multi-Omics Data Integration for Cancer Patient Stratification konulu bir konuşma gerçekleştirecektir. Merhaba hocam, hoş geldiniz.
Merhaba, hoş bulduk. Sağ olun. Teşekkürler davetiniz için ve organizasyon için.
Rica ederiz. Dilerseniz sunumuza başlayabilirsiniz.
Tamam, ben yani duyuru İngilizce olacak diye yapıldı. İngilizceye geçeceğim. Yani sanırım pratik olması açısından da faydalı olabilir. Bir de ben şimdi bunu Türkçe sunarsam muhtemelen yarısı İngilizce olacak. O yüzden İngilizceye geçiyorum şimdi. So I'll talk about this how to do multi-omics data integration for cancer patient stratification. So first I will need to, of course, convince you why we need to do multi-omics data integration, and of course need to give you some background. So first, this is the outline of the talk. So basically...
Affedersiniz, çok özür dilerim, bölüyorum ama sunumunuzu paylaşabilir misiniz acaba?
Sunum şu anda paylaşılmış durumda olması lazım. Tam ekrandayım ama.
Şu anda bizimle paylaşılmış durumda değil hocam.
Nasıl yapacağız?
StreamYard'da da bir tekrar paylaşmak
doğru, pardon. Tamam, doğru, pardon. Başa alayım.
Estağfurullah.
Şimdi görünüyor mu? Tam ekran olması lazım.
Evet, harika.
Tamam.
İyisinler hocam.
Tamam, teşekkürler. So, we need some, I'll try to give you some background in cancer biology, and it will be a bit intertwined with why we need multi-omics integration to study cancer, to understand cancer. And because I'll do some exercise in R, I'll need to do also, maybe I thought I would give some quick introduction to machine learning as well, how it works. And then, because it was a bit hard to, because I don't know the audience, and maybe the audience could be students, or I don't know, PhD students, postdocs, could be professors, or people from all sorts of backgrounds could be the audience. So it was a bit hard for me to gauge, to organize the slides in that sense. So I tried to give a bit of an intro about different things. So hopefully everybody gets a bit of something out of this talk. And with the exercise, I'll do some multi-omics data integration with gastrointestinal cancers from TCGA data. And I'll show some drug response prediction in cell lines from CCRE database. So yeah, I'll try to cover as many applications as possible. So yeah, hopefully everybody finds something interesting from this workshop. So first, some background in cancer. So why do we study cancer? Why do we care about cancer? Because yeah, it causes so many deaths every year. 10 million people die. Just in US, it costs a lot of money, at 80 billion. It's the estimated cost just in the United States. And it's the cost of developing a single cancer drug is going up really. And then it costs a lot of money and time and human effort, of course. And not to mention, of course, the misery people go through, of course, the patients. So yeah, it's basically too many deaths, too expensive. It's a big burden on society in terms of quality of life. And yeah, everybody these days, the cancer is a popular disease, right? It's everybody, somehow you know someone who had cancer, and the media is always covering cancer stories, and so it is a big place in the media, but it's not a new disease. Actually, it's not a modern disease. It has been there for a long time. We can trace it back to 5,000 years ago, and probably, and it's not that young as well. It's probably it existed back when multicellular life evolved. Probably, I mean, when there is multicellular life, probably there was cancer. But yeah, now, but the struggle of humans against cancer can be dated back to 5,000 years ago. And in the beginning, it was just surgery. Like the available treatments would be surgery. You just remove the tumor from the body somehow, and maybe you would remove too much sometimes, probably. And think about the times when you didn't have anesthesia and stuff. And then we had radiotherapy in the beginning of 20th century. Then there came chemotherapy, some targeted therapies, and then latest these immunotherapies. That basically, as you go over time, the cancer treatments evolve and then get more and more specialized. We understand more and more about cancer, the molecular biology of cancer, and then you you get more data from more and more patients, and then you have better understanding, and then you develop treatments, better treatments, more targeted treatments that won't be as impactful on the quality of life. For example, when you receive chemotherapy, it's the basic function of chemotherapy. Basically, it will put so much pressure on the body that the idea is that for fast-paced cells, the fast-dividing cells are less likely to survive, like the cancer cells are really highly fast proliferating. They are less likely to survive than normal cells that are more robust against this kind of effect. But when you get the chemotherapy, of course, there are other cells in your body that are dividing fast, like skin cells, I don't know, hair, like that, this kind of tissues will be affected. But with more targeted therapies, the quality of life can be improved. And the most recent breakthrough was after understanding that the immune system actually is very important in attacking cancer, of course, but then it was found that the cancer cells develop mechanisms to evade the immune system. So the immune cells they're always patrolling the whole body, and then cancer cells, when they become enough different from the normal cells, then they become targets of immune cells. but sometimes the cancer cells will develop methods to avoid this kind of immune cells being prey to immune cells. So there are therapies that will basically fix this problem, the immunotherapies. So if you'd like to learn more about this history, the background of these things, I would recommend going and checking out these books. They're very good books about. So the first one on the left is about the history of cancer. The one on the right is about the breakthrough invention of immunotherapies. Of course, none of these treatments work perfectly, and still, I mean, all those times, all these years of struggle against cancer, and for a lot of the cancer types, we still don't have good survival rates. Some of them really survival rates are really low. Like pancreatic cancer is really aggressive. But even though, like, let's say you have a good survival rate for some cancer types, still, as I said, they are not very. The treatments aren't very precisely attacking the molecular mechanism of action all the time, and not everybody also benefits from every kind of treatment, and everybody is unique in their way of developing cancer. So this not surviving cancer is of course great, but also maybe you want to not lose a body part, for example, after when you survive it. So this kind of, so what I'm trying to say is there's still a lot of room for improvement in many different cancers, and for cancers that the survival rates are good, still you can improve the quality of life. So why I mean, despite all the money, all the effort, all the misery that's gone into cancer fight against cancer, why do we still have not perfect treatments? Why don't we have this? I mean, if you ask someone on the street, maybe they will say, okay, it was the big pharma, they want to profit a lot on this, and they don't want to tell you that there are treatments, or they don't want. I don't know why they wouldn't, but they would make money if they had treatments for all of these cancers. I think the basic misunderstanding is that people don't understand the molecular biology of the disease, of course, because it's not one disease, it's hundreds of diseases, different kinds of diseases, and they occur because of different reasons in everybody. And it's hard to understand this without putting enough effort and money and research on it. So first, I will just give you some contrast with the Mendelian diseases and cancer, to illustrate my point. Basically, with the Mendelian disease, for example, the sickle cell anemia. With the normal red blood cells, you have this hemoglobin, and then sometimes some people will inherit a disease mutation, a mutation in this hemoglobin gene. It will just change one amino acid to some, like a glutamine to valine, and glutamic acid to valine, and then this substitution will create problems in the structure of the hemoglobin, and then you will have this instead of having these normal round-shaped normal red blood cells, you'll have this sickle-shaped red blood cells. So what's the problem here? The problem is these abnormal red blood cells cannot contain as much oxygen around. So, and then you need oxygen to survive, right? And then, so that's the problem with this disease. And then let's say another monogenic disease, a Fabry disease, where you have a problem with the enzyme. There are mutations in this enzyme, and then when the enzyme mutated, it's not properly localized in the cell. And then it has all these different phenotypes in the people. So it's an inherited disease. You'll have these mutations in your enzymes. If the enzymes doesn't function properly, you have all these different symptoms. And yeah, it's a rare disease again. But what I'm trying to illustrate with these examples is that with these kind of diseases, you have inheritance. They inherit from your parents, and then you have simple changes in the genes or mutation of maybe the problems in the genetic component of these diseases is that you have fewer issues, like things to consider, like maybe one gene, maybe one mutation, maybe different mutations of a given single gene. So I'm not trying to say that these diseases are easy to treat, but what I'm trying to say is that understanding them, or maybe finding treatments for these things, would be the genetic component might be a bit simpler. In the sense that you can focus on one gene and then try to fix that, maybe. For example, with the new genetic modification technologies, genetic editing technologies, maybe we'll have this kind of simple treatments later. But with cancer, the story is different. With cancer, so what's cancer basically? Cancer is uncontrolled growth and proliferation of cells. And if you think about at the conception. We are just one cell, right? At the conception, the zygote. And then it divides and then proliferates and then changes and differentiates the cells as it grows. It changes into different tissues, structures, and then we have this big organism with trillions of cells. And so this requires a lot of division, differentiation, and a lot of cells die, and a lot of cells are renewed, and every day you'll have billions of cells dying, billions of cells coming up again as new. During this process, for a cell to with cancer, basically some of these cells might lose their the control. There are a lot of checkpoints for a cell when it differentiates, or let's say your skin cell will be very different from a muscle cell, although it has the same genome. That the way it's supposed to be functioning will be different from different tissues. But sometimes they will lose some of the cells will lose their way of interpreting information, signals coming around, or they will kind of declare some kind of independence from their surroundings, and they will basically have this uncontrolled growth and division. And if they can divide enough, then they form these tumors. Maybe they will metastasize to other places. So, but the bottom line is that for cancer to occur, some of the cells need to lose this kind of control and turn into proliferating cells. But the problem is this can happen through many different channels. The example that I gave here with these diseases, you would need one gene affected for the disease to occur. But for cancer, you have many more different channels to happen. So, and these are the hallmarks of cancer. There are many different ways a cell can lose its control. And each of them is a big research field, of course. And these hallmarks keep getting updated as we understand cancer better. And of course, and cancer is also not something that's, okay, some cancers are inherited, maybe easier to understand and treat, but a lot of the cancers are not inherited. They happen later in life, maybe due to your interaction with environment, or as I said, it's a disease of the cell. Like you have this, when you look at a tumor, and it's called, this picture depicts the microenvironment of the tumor. When you look at it, it's a bunch of cells. Some of them are cancer cells. Some of them are your immune cells who are trying to kill your cancer cells. Some of them are blood vessels, again, your normal blood vessels, or some blood vessels that are recruited there so that because of cancer cells signaling. Because they need to survive, they need the cancer cells, so that they can grow and proliferate, they need energy. And they need blood flow. So this disease is not just, and when you look at the microenvironment, every cell may have a slightly different genome. Every cell may have a slightly different molecular profile. And then when you take it, and this is not something that normally exists in your body, that the cells, the cancer cells, are different from normal cells. That's why the immune cells start to attack them, because they look different. They look like foreign cells. But within this environment, there's also all sorts of competition. All these cells are maybe the cancer cells in this area. By the way, can you see my mouse cursor? Mouse'um görünüyor mu acaba? Bir feedback
Evet hocam,
alamıyorum ama.
gözüküyor.
Tamam, pardon.
Mouse'unuzu görebiliyoruz.
Tamam, teşekkürler. Maybe if you pick a cell from this area and then another cell from this area, maybe they'll look different in terms of their biology, in terms of why they develop into this cancerous state. So even this tumor coming from the one person could show a lot of heterogeneity when you look into the microenvironment. So that's why it's a complex problem. So it's hard to distinguish normal from cancer, and even if you take a sample from a tumor, like you take biopsy, it doesn't reflect just pure cancer cells. So there's a lot of complicated stuff going on. So as I said before, so there is molecular determinants of cancer. There is also interactions between cells, and here it's not depicted, but there's also the microbiome that's very important. So our body is full of microbes that also help us with our fighting against other pathogens or processing food and stuff. So they also might play a role in this environment. So it's a complex disease. That's why it's hard to treat it. It's hard to find the problems, what's going wrong, and each person may develop it for slightly different reasons. And even for a single person, even given a single tumor, you might have variation that might make it difficult to understand what's going on, what's going wrong. I'll give you one example of how cancer treatment works in the clinic right now at the current state. By the way, I'm not an oncologist. I'm not a medical doctor. So hopefully I'm not saying something wrong. And please correct me in the comments if you are specialized in this area. But basically, the current state of diagnosis of cancer or treatment. Let's say take the breast cancer as an example. So when you have potential breast cancer, a tumor in the breast, and then go to an oncologist, some of the main things that to first consider will be the age of the patient or the gender, the menopausal status. The gender might be important because men can also have breast cancer. And then the tumor stage. The tumor stage means, okay, how big is your tumor? And how many of them do you have? Have they spread to lymph nodes around? Have they spread to other distant organs? So there is all sorts of things to consider that you can directly maybe like the macro features of a patient you can observe. Then there's also genetic determinants of how you would treat a patient or how you would diagnose a patient. Like, for example, there's these three important receptors. There's epidermal growth factor receptor, estrogen receptor, progesterone receptors. These hormone receptors are important biomarkers for deciding what kind of therapy to give. For example, if you have high expression, high activity of these receptors, you might want to give hormone therapy. And there's these other genetic determinants, like if you have mutations in your BRCA1 or 2 genes, there are other inhibitors that are designed for this kind of problem. Recently, there's these immunotherapies that are developed for immune checkpoint inhibitors, basically that depending on the status of the activity levels of your some of the genes that are proteins that are important for the cancer cells to evade the immune cells. You can use some kind of inhibitors to break this interaction. And there's also multi-gene panels. For example, there are these companies that provide these panels that contain 50 to 70 genes, basically looking at your transcriptome, the gene expression status of these genes, and then basically try to understand if this patient is likely to benefit from chemotherapy. Of the chemotherapy, let's say you took a chemotherapy, would you be likely to have a recurrence of the disease or not? So these kind of things can help. So here, I'm not trying to give you a lecture about how to treat breast cancer, but I'm trying to say that the molecular determinants that are currently considered in the clinic are just few things. Like, okay, you have few receptors, maybe few genes, maybe 50 to 70 genes. Maybe compared to the clinical practice, of course, there's maybe a lot already, but I'm trying to make into that point that maybe we could do even more than that. And yeah, for breast cancer, I mean, I'm not trying to downplay. So this is all these developments. Of course, this is great that all the struggles, all the effort people put into researching this disease. Now, I mean, compared to 40 years ago, these are statistics from UK that compared to 40 years ago, people can survive cancer twice better. So basically, like for breast cancer, it's 80% survival rate for compared to 40 years ago, which was 40% at the time, because you didn't have all these different options that would cover a bigger proportion of the population that might develop breast cancer. And then there is until this point, what did we see? Maybe we saw up to 50 or 70 genes that might be important for clinical diagnosis or treatment guidance. And now we are also more and more seeing in clinical practice or some places that there are these cancer gene panels that from these different from Foundation Medicine is a company that, and then there's OncoKB from MSK, where you have up to maybe hundreds of genes that they have these panels that are associated with cancer that are important that reflect the biology of cancer. And then from these panels, we can do some sequencing, and then basically you have an understanding of which cancer-related genes are mutated, which ones are maybe amplified or deleted. like copy number alterations. And then you can have some derivative features like microsatellite instability, tumor mutation burden. These are some of the factors that to take into account when kind of grouping patients in terms of, okay, which ones are likely to benefit from certain therapy. For example, patients with high mutation burden are more likely to respond to immunotherapies, some of the immunotherapies. And that's also, of course, depends on the cancer type. And it's just general markers that people are trying to use for diagnosis and treatment. But still, we made it to maybe 700 genes, and maybe we look at mutations, copy number variations, but we are still looking at the genome and maybe partly the transcriptome, but a small proportion of it. Like with the genome, we know there's more than 25,000 genes, but we are maybe looking at hundreds of genes, and maybe transcriptome again. We have maybe hundreds of thousands of transcripts, but we are looking at a few of them. And this picture is basically like a depiction of what kind of reflects the human health condition. It's from Eric Topol. I'll also recommend a book from him that this basically the human health, although the genome is already a good indication when you look at the genome, maybe some of the markers in the genome that will help how to be treated for a given disease, it still doesn't reflect the complete picture. Then what the human health condition for a person to respond to a certain treatment, there is many things that can contribute to this process. Many different layers of information, like the genome, the transcriptome, the proteome, the metabolome, microbiome, epigenome, exposome, and like even if like these are the first layers I counted are mostly molecular determinants, except the microbiome. But then there's this environmental factors, like the kind of food you eat, maybe the kind of exposures you get. And then there's additional kinds of layers of information you could use, like medical imaging, some wearable technologies that will help you maybe profile some of your health status. And I don't know, the geography you live in, the climate, there are all sorts of things that you can contribute. Like there is all sorts of many different layers of information one could use to assess the health condition of a person. But currently we are using a very small proportion of it. So that's the thing. So that's why we need more understanding of these different layers and use them more maybe in the clinical practice. So I'll try to give you a bit more, a bit of the, how these different layers are affected, maybe how they can be used in how they're relevant for cancer. For example, with the genome, okay, or it's maybe the most well-known, you have mutations, small mutations. You have, you maybe let's say this is a normal chromosome, then a chunk of DNA gets inserted in it, or a chunk of DNA gets deleted, maybe it gets inverted, it gets duplicated, maybe you have a chromosome and then part of it is just removed and spliced back into another chromosome. And then the same chromosome, you have a segment of DNA that gets duplicated. Then you have multiple copies of the same junction of the same segment. So these things will, of course, impact the whole genomic content, because I mean the genes that are in the DNA, they are not just randomly distributed. They have interrelations. The genome is packaged in a way, and it's cell type specific in a manner I will talk more about in the epigenome introduction. So these things, these aberrations, these damages in the DNA will have different outcomes, and these can be different from person to person. So that's why they will have different effects on each patient. And then there's these catastrophic events in the genome. Like you have this chromosome that's completely, it's something called chromotrypsis. You have these completely exploding chromosomes and then bits and pieces, and then the cell tries to repair them and put them back together, like solving a puzzle, but it doesn't work properly because the damage is too much. And then so cancer cells may have this kind of catastrophic events as well. So what else? So we have another layer, the transcriptome. Let's say everything is fine in the DNA, but and then this is just quick crash course on molecular biology, basically, how the how we go from DNA to proteins. So you have the cell, and then you have a nucleus, you have the DNA, then there is a transcription machinery that goes and sits on top of the gene and then transcribes the mRNA molecule from it. Let's say this, we are focusing on messenger RNAs that are later translated into proteins. And then you have a bunch of complexes, protein complexes that need to process this premature mRNA that will process it. Like, you know, there's introns in the genes that needs to be spliced out, and then there needs to be some additional decorations on the transcript to make it more stable, and then like this polyadenylation or 5' capping. Then you have this export of this transcript to the nucleus where it gets translated in the ribosomes, and then maybe once you have enough of this protein, maybe you need to degrade the transcript, and then the adenylation, degradation. So there is a whole sorts of so many different steps and so many other players in this complex processes, and at each step there is some tight regulation that needs to happen so that you prepare you transcribe the right gene at the right dosage, you splice the right introns, the right places, maybe you had the proper decorations around it, you translate it at the right amount so that you don't overexpress a protein. Let's say, for example, if you remember about the breast cancer example, you have these estrogen receptors that are produced way more than normal people, and you have these cells with so many estrogen receptors, and then the cell will be very receptive of estrogen hormone, and then the cell will always want to divide, keep dividing. So so you don't want this uncontrolled, high dosage or low dosage. Sometimes you want to have like there are these guardians of the genome that some proteins are important to degrade the proteins that are not supposed to be high dosage. So there's all sorts of complex interplay between within the cellular machinery to keep things in check, to have a proper functioning cell. And it divides when it needs to divide, it grows when it needs to grow, and it dies when it needs to die. So that's needs to be regulated properly. And those things you can see, you can all sequence like this, all different aspects of the transcriptome, and then have a kind of a picture of how a cell is doing. And then you have the epigenome. Like epigenome basically is epigenetic mechanisms are what actually makes us go from a single cell to a complicated organism with trillions of cells, but still have a function properly. And so like when you have a single cell, with a single cell, you start with the same genome that yet gets copied to every other new cell that's generated. They all have the same sets of chromosomes, hopefully, but each cell functions differently. Like your skin cell is different from your muscle cell. How does it happen? Although they have the same chromosomes, they have the same DNA. Why do they have different types, different shapes, different functions? So that's kind of regulated at the epigenetic level. So and the DNA within a cell is not like this open, sits in this open conformation. It's always like it's packaged, and then it's maybe if you zoom in to a gene that might be accessible, that might be transcribed, maybe you have this. So you have the DNA sequence that's wrapped around with these histone proteins, and then some places the genes might be accessible depending on how tightly packed the DNA is, and some places the gene might not be accessible, so it might not be possible to transcribe those genes. And so the structure, the conformation, the packaging of the DNA is also very important. So, and how is it regulated? Again, this is regulated by modifications on these histone proteins, for example, or modifications that are made on the DNA sequence itself, like DNA methylation or histone modifications. So if you look at every type of cell from different tissues, they will have different kinds of genes that are being transcribed, that are accessible, or depending on the signals that are coming from outside, the cell might decide to open up a certain part of the DNA, transcribe the genes in that part, and then pack it up again. So all these things need to be regulated at the epigenetic level. And even if, let's say, you have the same DNA that's perfectly in your normal cells and in your cancer cells, maybe the cancer cell has a way of packaging the DNA differently, that or it is the packaging doesn't work properly, that some of the genes that are not supposed to be accessible all the time might become accessible all the time, and then those genes might contribute to growth and proliferation of these cells. And then there's the proteome, right? If you go from epigenetic mechanisms, then you have the genome. Maybe you have 25,000 genes in the human genome. Then you have these alternative transcription events, splicing. Then I talked about all these mechanisms, and the RNA might get edited over time, and then you have these richness, novel richness, the complexity that is added on top of the initial layer, where you have the DNA, but from these 20,000 genes, you might get 100,000 transcripts, and from the transcripts you get proteins, but each protein from each transcript might be, again, decorated differently. You can call these are called post-translational modifications. Like you have these proteins with additional chemical moieties that are called basically like ubiquitinylation, sumoylation, phosphorylation, acetylation. All these different modifications that are put on top of protein sequences affect how a protein is folded, how they interact with other proteins, how they are regulated, how they are localized to different parts of the cell, how they are degraded, for example. And then that affects basically all these different complexity through these modifications can give you a pool of different kinds of proteins that will have a different composition. And so again, this complexity can be slightly different from cell to cell and from person to person. And with cancer, again, all the way from here to here, different kinds of things might be going wrong. And then the total outcome of this whole thing can be, okay, let's keep growing, let's keep proliferating. And that could be the thing that contributes to cancer, and differently from person to person. And all these layers I mentioned are just layers that that are at the molecular level within a cell, right? And as I said before, if you look at the tumor microenvironment, you have all these interactions between cells. So at the cell biology level, we need to understand how cells function, how they interact. And like, for example, the immunotherapies will work with the principle that, okay, some cancer cells can develop some mutations or epigenetic mechanisms or some differences in proteome that will lead them to be resistant or to be able to evade the immune cells, so that they are not prey to the immune cells, and then they can survive all the treatments that we do. Maybe they can survive all kinds of stuff, all kinds of treatments and immune system. That's why, okay, the immunotherapies have been found to be breakthrough therapies, but still a big proportion of people still don't benefit from it, because we don't have treatments for everybody, because everybody has different kinds of cancers. Yeah, and then yeah, I guess I'm kind of repeating myself, trying to emphasize the point that the health status of a person doesn't depend on just one layer of this information. And if you put together all these layers, you end up with having millions of different features, and then it would be difficult to find two people with the same molecular profiles, right? Or to say with the same measurements. And why don't we just do this? I mean, okay, there's all sorts of information you can get that you can use to understand cancer, but why don't we just do it? It's because it's economies, right? It's expensive, it costs money, and you need to first have enough number of patients to study, and they should have enough number of. They should be hopefully have some unique common problems that you can maybe study and develop some drugs that will fix all those common problems. So it's an expensive endeavor, basically. But now there's more and more. It's getting cheaper to do sequencing. Like you can have for all these layers, maybe you can generate data much more cheaply than maybe 10 years, 15 years ago. There's an exponential decay in the amount of money you need to spend on per sample for sequencing. And then there's more explosion of data due to this cheaper sequencing prices. And then, of course, when the data explodes, you need more computational methods to make sense of this data, and which is also a good time because we have more and more methods for doing machine learning, so that we can handle all this kind of different big volumes of data. So, bottom line is, the summary is, if you, let's say you have this, you studied all patients you could study that have this cancer, and then you had a map of this, all the patients. Let's say every point is one patient, and then you had a map of them, and then okay, you can decide okay which cancers they have, and then maybe you can zoom into some of them. What you can see is that it will be hard to find two patients with identical profiles, but you can find groups of people with similar problems. So that's what means that that's the mission of precision medicine. You try to segregate, stratify patients so that you can group them with similar problems, similar molecular reasons, or environmental reasons, or whatever data you have that leads to this state of cancer. And the purpose of this stratification can be different things. Like, for example, you want to find which group of patients is likely to respond to a certain drug, which group of patients are likely to survive the best. Maybe that will help you decide on what kind of treatment you will give. And then subtyping of cancer, like you try to come up with groups of similar patients with similar underlying molecular mechanisms that are going wrong. And then, like for example, you want to figure out, which patient is more likely to metastasize, or which patient's tumor is more likely to metastasize, because metastasis is the biggest reason for death, for example, from cancer. And let's say you zoom into one cancer, you collated all data you can have, and then you found that you come up with this map of patients for a cancer type, and then each patient each point represents one patient, and then what you can do is that maybe I'm trying to illustrate the points that I just made. Like maybe this group of patients, if you look at this map of patients, maybe this group survives the best. Maybe this group responds to a treatment A the best. Maybe this group of patients respond to treatment B the best, and maybe this group is a bit of more unlucky that there isn't any treatments and they're not likely to survive so much, and then they're most likely to metastasize. And let's say a new patient comes, and then hopefully you can use all these different information you can use for this patient, then put them on this map, then maybe find out hopefully this patient is in this place where there are multiple treatments available, and then they're likely to survive this cancer. So hopefully I kind of gave enough background on cancer biology. Why do we need to do stratification? And now you need some tools to do this. And that's why we have all these multi-omics integration tools. And so why do we need multi-omics integration tools? So first of all, as I've been arguing, that we are not using all the information we could use to understand cancer and treat cancer. And then we have all sorts of these different assays like transcriptome, proteome, epigenome. We could measure different things, but they all come with different sizes. Like maybe with the proteome, with the transcriptome, you measure tens of thousands of things. With methylation, let's say epigenome for methylation data, you measure maybe hundreds of thousands of features. With the proteome, maybe you get thousands of features. So they all have different sizes. So we need tools that can somehow, and also they have different distributions, different kinds of distribution within each assay. So you need to be able to somehow put them together and put them into the same space of distribution and with a common scale, basically. And then the other thing is maybe I could show you this picture that, okay, these are all known, maybe the major pathways in cancer. And if you look at it in this two-dimensional space, what the different patients will have different problems with any of the genes in this place, but then they'll also different layers of this that we can measure will be affected for each patient. So these tools will help us somehow find out these interactions between different layers, and then come up with a better representation of these maybe correlated features. Let's say there's this gene that is mutated in one patient, and then this gene gets overexpressed, maybe, or another patient has an down regulation of this gene, maybe, or because of epigenetic reasons, and another patient has a problem with this gene because of some post-translational modification problems. So the multi-omics integration tool will somehow will be able to put together these different kinds of information, because these layers actually don't live in, I mean, this information like proteome, transcriptome, epigenome don't live in separate compartments in the cell. They are all happening within the cell, and they all interact with each other. So so what we need is to extract meaning, common meaning, out of all these different issues that lead to a similar outcome from all these. Let's say if you think about this like a computer circuit, and what happens if you break this part to this computer? Can it still function, or maybe does it still stay on all the time? Maybe you can't turn it off. So there is a lot of multi-omics. Okay, and then one more thing I forgot to say is that, as I said, there's tens of hundreds of thousands of features you could measure, but this is a big problem in to make sense, because this is something called the curse of dimensionality, where maybe you have hundreds of patients that you could get to study a specific cancer if you're lucky, but you have hundreds of thousands of features to study that you measure. This is very difficult problem to make sense. So you need to somehow reduce the dimensionality of all these different features to a more manageable and meta features, they could say, or latent factors that they are called different things based on different tools that will reflect the overall meaning of what's happening in the cell. I would recommend these two review papers to go and check, and then maybe download some of the tools to multi-omics integration. Then you can see differences between what they do and then how they work. I'll give some examples here. Like some of the tools can be just clustered in this part. And one of them is actually developed in our lab, MAUI. But I will show an example using MCIA today in the exercises, because it's simple and easy to install and then run. So there is a dimension reduction aspect of multi-omics data. As I said, there's too many features, and you want to reduce the dimension of the features while catching the essence of the data, keeping the essence of the data. It's like compressing. Let's say you have a music file, you want to compress it, but still be able to make sense of the music, enjoy the music without having problems. Without you still the music file doesn't take too much space, but you capture the essence of it. Or there's some tools that are also more focused on subtyping cancers while reducing the dimensions. And there's some tools that just are focused on clustering and subtyping. They don't do this kind of, they won't give you this kind of reduced dimensionality meta features. But like these dimension reduction methods can be more flexible, because you reduce the dimensionality, you have these useful features that are, you have fewer features than number of samples, and then you can use them for any sorts of machine learning things. Like you can do clustering, subtyping, and all sorts of classification or regression tasks, which I will introduce later. Hopefully I'm not spending too much time. Okay, okay. I'll try to speed up a bit, because I want to do some exercises as well. It's also a bit weird, I mean, doing this kind of lecture, because you don't get to interact with the audience so much. Hopefully everybody is following what I'm saying. So overall, what's happening for these dimension reduction methods is kind of follows this logic basically. Okay, they're not the same, they are slightly different, or different methods will have different ways of doing this, but the basic idea is that you have these different assays from different people, and these assays may, let's say you have macro transcriptome, proteome, I don't know, genome, and then they all come in different shapes and sizes, and you have the features and then samples, and you want to find you want to reduce these matrices into this simplified matrix of latent factors or meta features, as I said before, that reflect the essence of these different layers, plus how they interact with each other. And then also you want to be able to have access to how these different features from these different layers contribute to each of these factors. So later you can go back, let's say you find a meta feature that's important for survival, you can go back and then find out, okay, which features contribute most to that factor, and then make more sense of it. To understand the molecular biology, maybe there's a gene that interacts with the protein in terms of at a different layer. Those things you can go back and figure out. And there's this integrative clustering methods that I will show an example. The similarity network fusion method that I like, that I'm just showing as an example. Let's say you have different layers again of omics data. What this tool does is it will form this network between each patient, each pair of patients, based on these molecular profiles, and then do it for different layers, and then come up with a consensus network. And all these different layers will contribute to the network, and then in the end, you try to find the most tightly connected communities within this network. And then the premise is that the idea is that using individual layers, you don't get as good a separation if you use the integrated version. Like if you look at this heat map, you get you can see the structure of the similarities between patients much easily than these individual layers. That's the idea of integrative clustering. Then there is also these deep learning methods that will make sense, make use all these different. This is developed in our lab actually by a colleague called Yona. And so again, the idea is that you can use these deep learning methods to feed these multiple layers of omics data. And there's this special architecture called autoencoders in deep learning. And that will, maybe I don't go into detail here because it will take additional time. But the idea is that you want to compress the input in this bottleneck layer, and then so you are able to decompress it in the end. So you can reconstruct the input with just using a smaller proportion, like the reduced proportion of reduced dimensional data can be used to just decompress it. It's like zipping files where you can zip a file and then you can uncompress it again, and you have the actual file again. But so that's why the zipped version, the compressed version, still keeps the essence of the actual input. And we also use this. Recently, we wrote another paper, which is not published yet, but it's on bioRxiv. If you want, you can check it out. I will make all the slides and the code that I will use later available to everybody, so that you can later study and go through all these links. Hopefully, these slides and code helps you as a starting point. If you're new to the field, you can just use them as a starting point to learn about these different concepts, different applications. And how can we study cancer with multi-omics? Where can I find some data? I'll show some examples. One of them is the big project, the Cancer Genome Atlas project, where the big consortium, different labs from all around the world contributed to the project. You have 33 different cancer types, more than 11,000 tumor samples for 7 different omics types, layers for real human cancer samples. And then there's also disease models like cell lines, organoids, patient-derived xenografts that are engrafted on mice that you can study. There are all these models also you can do a study with the multi-omics. So there is one, there's these cell lines that I'm going to show one example from the CCLE, Cancer Cell Line Encyclopedia, how to do drug response prediction. So in this database, they have they basically took these data sets, did multi-omics on each cell line. So for more than up to, I guess, more than 1,500 different cell lines. And then they treated them with different drugs and measured how much each cell line responds to different drugs, and then measured them. And then basically you can do, later you can study this using multi-omics features to predict how each cell line responds to drugs. And then if you can get some understanding from these cell lines, then you can translate them to other better models like mice and then humans in clinical trials. So I'd like to do a quick introduction to machine learning as well, because when we do multi-omics integration, we'll obtain these latent factors, and then we'll use them to do more things on them, like subtyping and survival modeling or drug response prediction. And then how do we do that? There's a workflow in machine learning that you can basically apply to not just biological data, but also other kinds of data from other fields as well. So these skills that if you get these skills, you can also transfer them. Let's say you are trained as a bioinformatician, then maybe you can find a job in, I don't know, booking.com or something, or some bank. You can do finance, because a lot of the skills you learn can be transferable. All you need is basically, with the machine learning, is basically that some measurements and some outcomes from these measurements for these different samples. So if you can reduce your problem down to this kind of picture, where you have samples, hopefully a lot of them, and then you have some features on the columns that, let's say, as I said, you can use multi-omics features, or you can, let's say, this could be a problem in finance. Let's say you are trying to understand which customers are likely to pay their credits, pay back their debts, for example. And then the outcome variable basically could be, let's say, in finance, okay, which customers paid back their debts on time. And then the features about these customers could be about, okay, the age, maybe the income, and all sorts of different features you could find. Again, the same idea with the multi-omics. You have samples, let's say tumor samples. You have different genes, maybe proteins and transcripts, and then you have some outcomes. And depending on the outcome could be, okay, for example, how long is a person going to survive? Some kind of outcome. Or how long is a person is going to respond to a treatment? Is a kind of outcome. What kind of cancer does this person have? Subtype of a cancer, for example, is another outcome. So depending on another feature that you want to, another label that you want to be able to use, so that you can understand the differences between, so you can build an understanding of which kind of features are most important in grouping samples or classifying samples or predicting some numerical outcomes like drug response for different samples using these different features. So if this outcome variable is a categorical variable, let's say A or B, it's a classification problem. If it's a numerical thing, let's say it goes from 0 to 1 and there's some drug response, then it's a regression problem. If you don't have any labels, then you can do clustering. So this is basically the whole grouping of how machine learning works. And so you can do clustering and then maybe group them separately and then figure out which different groups, what kind of features they have different from each other. Maybe here I can say, okay, in supervised learning, that means where you have labels of each samples, you do classification or regression where you have these dots and then you know what they are, and then you try to learn using their features, you try to learn how to separate them from each other or how to predict some numerical outcome. Or if you don't now have any labels, then maybe you can cluster them in an unsupervised way using the features that you know, that you measure. And then maybe, okay, this group seems to be different from this group. And then maybe you figure out, okay, this group has a high level of this gene and this gene, but a low level of this gene and this gene. So this kind of grouping helps you figure out, okay, what kind of features are could be useful in figuring out grouping patients. So, and this is the whole workflow of, maybe I won't spend too much time on this, but of how machine learning works. You have the initial data set, you do some exploratory data analysis, some data cleaning, then you get this matrix, pre-processed data. Then you try to, you want to split this data into training and testing sets so that you train on this model, so you don't, in an objective way, so that you don't, your model can work in samples that you haven't yet seen. Like basically, like the test set serves this purpose. You don't look at the test set. You do everything you can with the training set and train all these different algorithms and come up with a train model. And when you use this train model on the test set, you do some predictions. And then with the test set, you can check if the predictions that you did actually make sense, if you evaluate them. And if it's a classification problem, then you have different metrics for evaluation, like how accurately you are predicting the labels, how specific or sensitive is your model to a data set that you haven't seen before. Or when you do a regression model, let's say you predict drug responses for the test set, then you can check the correlation between your predictions and then the known labels. I'll quickly introduce maybe I'm spending too much time, but it's important to go through these topics as well, because when you do the exercise, it will be important to have these intuitions of how these different methods work. I will use random forests in the exercises, but before going into random forests, I'd like to give you an introduction to decision trees. So this is one type of algorithms that you can use to train a model on different features. Let's say you have these samples, and then you know their labels. And then based on gene X and gene Y, this is how they are clustered. And how can we build some kind of procedure? Let's say when you have a new sample, can you tell, is it more likely to be an A or a B or a C? So we can already visually see, I mean, it's easy for us to do it. Like there's only 2 dimensions. We could do a split in the middle. Let's say anything that has a gene X lower than 6 probably belongs to group A, and anything above 6 is more likely to belong to B or C, but we can't tell if it's B or C just by using gene X. So then we can do one more split based on gene Y, and then maybe that for samples with gene X higher than 6, we could also look at gene Y, that maybe those that are more than that have a Y value of above 7, that we can call them a B, and then if they're lower than 7, we can call them a C. So then you have this kind of a tree structure, like decision tree. That's why you call it decision trees. When a new sample comes, you check their value of their gene X, then check if it's above 6 or below 6, and then then you check gene Y, if it's above 7 or below 7, then you decide if it's A, B, or C, based on just 2 features. But the thing is, in real life, as I said, there's tens of thousands of features to consider. We can't just look at it in 2 dimensions and say, okay, let's do this split, And there's these random forests. Basically, it's called forests because you build many of these such trees. Like on the left, you have a decision tree for a simple 2-class classification, and then you make many of these things. You build many of these trees, hundreds of them, maybe 500, 1,000 of them, and then each tree will consist of different features, and then they will be randomly selected. That's hence the name random forest. So you'll have a random subset of features, and make a decision tree, and then do it many times. And then from the labels that you know already, you can train a model. And then from all these decision trees, you'll have certain outcomes. Okay, some of them will vote. Okay, this is the new sample is likely to be class green, or some others say, okay, it must be class red. But maybe doing a vote across all the trees will give you a single outcome. The random forest will tell you, okay, this is most likely to be class green or red. And then the random forest will use, because it's simple, easy to run, and also they are quite robust. And you can use the same training procedure for regression or classification problems. So yeah, here I'd like to say that if you'd like to learn more about machine learning, statistics, and some other omics analysis methods like RNA-seq, ChIP-seq, isoform sequencing for methylation analysis, or multi-omics integration, I would recommend this book that we recently published. So most of it is written by Altuna, who's the head of Bioinformatics platform, and my colleagues and I, we contributed chapters to this book, and so you can also find it freely on this link. So you can also buy it on Amazon, but yeah, you could just do it on your own. You can do it in our it's available online. And I'd like to also quickly recommend some books that when I talk to, I hope that people recommended me a lot of books before that I could read about science and all the things that are relevant to what I just talked about today. So some of them, so the ones on the below row, the bottom row is that, so you could use those books to learn about how to use Python or R for programming, machine learning, and applying them on data, not just multi-omics, but other kinds of data from other fields as well. And on the upper row, we see books about, okay, the history of cancers. You can learn about, this book talks about the first cell line, cancer cell line, HeLa cell line, maybe you know about it. It's actually named after, the name comes from the person who had this ovarian cancer, and the sample was derived from this person. And then this immortal cancer cell line was developed and used all around the world. If you want to learn more about immunotherapy and cancer, I recommend this book. If you want to learn more about how artificial intelligence will change how medicine is practiced, and there's also a chapter about multi-omics in this book, this Deep Medicine by Eric Topol. If you want to learn more about the history and also how epigenetics is important, I would recommend this book. This book from Carl Zimmer is more about the history of genetics and heredity, and so that's also would give you some view of how we came to this point of using molecular information for all sorts of things. And then this book is about the microbiomes that in our body, you know, like most of the cells in our body actually belong to microbes, and they are very important for our health. And actually, a small proportion of the microbes are dangerous for us, but we kind of, when we say microbes, it doesn't have a good ring to it. It must be something, microbe must be something bad, but it's actually, they have a lot of important features for us. So yeah, I'd like to acknowledge the lab members, and also, yeah, definitely the organizers of the conference, and Elif as well, especially. She was very helpful in organizing, and all the Bioinfocongress organizers. And yes, so maybe I can do exercises now. So if you go to this page on GitHub, you'll find the links to the slides and all the code and data that I will use to demonstrate the analysis. But now I feel like maybe I should get some questions, but maybe later. I don't know what the moderators think. Is there any questions?
Merhabalar hocam. Dilerseniz bir 10 dakika ara verelim, dinlenme arası. Daha sonra çalıştayımıza devam eder. En son soruları alabiliriz.
Tamam. Evet, bu tamam. Şeye başlayınca R'daki egzersizlere başlayınca şey yaparım. Ben yani söylemem gereken birkaç şey var. Burada yani zamanımız biraz kısıtlı olduğu için tamamını ayak uydurmanız zor olabilir. O yüzden mümkün olduğunca yani koda çok takılmayın. Yani kod nasıl çalıştı, nasıl oldu, ona çok takılmayıp mümkün olduğunca neyi neden yaptığımı anlamaya çalışırsanız, takip etmeye çalışırsanız 45 dakikalık süre içerisinde herhalde en azından bir genel bir fikriniz olması açısından veya neyin nasıl yapılacağı hakkında fikriniz olursa daha sonra bu zaten dediğim gibi materyal hazır olacak. Online GitHub'da bulabilirsiniz. Slaytlar da hazır olacak. O slaytlardaki bütün linklerden gidip konular hakkında öğrenebilirsiniz. O yüzden yani kendinizi çok zorlamayın. Yani koda yetişeceğim diye veya bu kod burada ne oldu, ne bitti diye anlamak için çok zorlamayın. Ama isterseniz tabii ki yapabilirsiniz. Kodu ve slaytları burada şu anda itibaren görebilmeniz lazım. Eğer sorun olursanız bana bildirin, düzeltmeye çalışayım. O zaman 10 dakika sonra mı?
Dilerseniz 5 dakikada olabilir hocam.
Tamam, 5 dakika sonra gelelim o zaman çünkü evet çok zamanımız kalmadı galiba.
Peki. Yeniden merhabalar hocam. Dilerseniz çalıştayınıza devam edebilirsiniz.
Merhaba, teşekkürler. Umarım faydalı bunu farklı uygulamaları içine çalıştım. Dediğim gibi kodu birebir takip etmek zorunda değilsiniz. Zor olabilir 45 dakika içerisinde ama daha sonra dediğim gibi bu şey üzerinden koda ve slaytlara ulaşabilirsiniz. Oradan daha sonra çalışmanız daha faydalı olabilir diye tahmin ediyorum. Şimdi kodu açıklamaya çalışacağım. Şu an ekranım görünüyor mu acaba? RStudio ekranı.
Evet hocam, ekranınızı görebiliyoruz.
Okay, I'll switch to English again to make it consistent. So, I think that the dependencies have been already shared with by the organizers. I apologize that I forgot one of the dependencies, but you'll find it in the script, the heat map library that might be useful to plot make some of the plots. So this here I'm basically, I will have to assume, because we don't have a lot of time, I have to assume that people know how to set up RStudio, install these tools, and how it functions, and some of the tools that I will use, like ggplot or whatever. So everybody might have to spend some time to understand how those things work as well. You can do tutorials. Each R package comes with a tutorial vignette to go through in more detail. So later you can spend some time for the things that you find interesting. So here I just run this command to set a theme for my plots that I will make. So I will just use this theme from ggpower and to ggplot2. And then in the folder you will find, okay, this code along script. Then there's these utility functions you can check out that I will use later here. So here I'm importing the functions that I will use later from here into this. So this line basically just shows, just changes the aesthetics, how the graphics look. Here I import the functions. Maybe I'll just do, first I need to, if you do command and enter, it will implement, it will run the code. And I just do these things. So in the folder, you should find a data folder where there's two data types, one from CCD, one from TCGA. First, I will start with the TCGA data. So this is how I will read the data, import the data. And this data basically is a list of different tables. And whenever you get some data from somebody or some database, you should do some first check the data, like how many samples do you have, like some distribution of the data, quick checks on the data set. And so in this data set where I have data from 4 different omics layers, copy number variations, gene expression, mutation, methylation. And this last one is basically a table of metadata about the patients. So the patients have these clinical features like that is saved there. So I will just like to put this in a separate variable here called call data. And then, so there's this script in the code that you can check. You can have a look how I downloaded data from TCGA. I use this script library called TCGA Biolinks, which you can use to download data from TCGA, all sorts of processed data and other kinds of like information about omics, drug information, I don't know, derived features and stuff. And copy number variations are coming from a different script. So okay, I won't go to too much detail, but yeah, you can have a look at those scripts. So the simple thing to do is, okay, I have these data types, but what is the dimensions of these things? Here, this function is I use to look at the dimensionality of these different things. So it's basically a loop. It's like a looping on each data type on the list, and then summarizing the number of rows and the number of columns. You see that it's the data, by the way, is I prepared the data. I did all the pre-processing. It's very clean data. That's the same number of features, the same set of patients, samples, and then there's this table called data with patient information. But in real life, of course, it won't be as clean as this, and then you will need to do some pre-processing. And let's first have a look at the patient-related data. So this patient-related data, there is information about the sample IDs, which kind of cancer it is, which kind of subtype it is, the age of the patient, where the biopsy of the tumor comes from, the gender of the person that gave from whom the biopsy was done, the tumor stage, histological types, and there is this specific columns for survival analysis. These are basically, this is called progression-free interval, basically in the number of days. Progression-free interval means you went to a doctor for cancer, and then you got maybe some treatments, and then you follow up. Maybe you go again to the doctor maybe 3 months later, and you check if your disease has progressed or not. And then some of them will have this information, like they will have some progress, some of them not. And then that's information that one can use to figure out, okay, some patients don't really have a progressive disease, but some do, and what are the differences between those? You can use the omics data to figure out. And then there is this, what you should do also, again, to have an understanding of the data, like what kind of, how many samples you have per, you can do a table command, how many samples you have per project. In this case, these are colon, esophageal, rectal, and stomach cancers. And you could check out the tumor stage. Okay, how many samples you have per tumor stage, and other kinds of factors. How many samples you have per rejection, per site of resection, and then what's the distribution of age? Like you can do this plotting histogram of the age of the different patients. You can quickly do those things. Maybe just check out the first table of the first 10 patients with gene expression data to have an understanding what kind of identifiers they use. Maybe, okay, let's have a look at the gene expression data a bit more closely. Okay, you have 1,000 rows and 300 columns, means 300. In this case, this is assay data, so people normally with machine learning, you should have the samples on the rows and then features on the columns. But with the omics data, usually you have too many features and then fewer samples. That's why you have these features on the rows and samples on the columns. So it's important to check where do you have, how your tables are structured. And then how many features do you have? How many samples do you have? And then some exploratory plotting, like I can use this core function from the stats package, which is comes with the R core libraries. You can compute, like this will do pairwise similarities between each patient. And you can also use this, you can visualize this, like you can do a heat map of these correlations between each patient. Like you can have a look, okay, there is this, maybe there is this group of patients that are very similar, and there is this big chunk of people, but there is within this big chunk, there is also smaller groups that might be more similar to each other than others. And if you do the same with methylation data, let's see how it looks. Maybe with methylation data, structure looks different. You see that with the methylation data, there is more and more smaller groups, but maybe it's easier to distinguish these smaller groups with methylation data. So as I talked about it before, that these different layers are complementary. They measure different aspects of the molecular profiles of patients. So they might have complementary roles in the understanding. So first, and okay, you could do a heat map, you could plot a PCA plot, or most commonly when you have too many samples, you have these methods called t-SNE, or you type t-SNE or UMAP, kind of the tools that are developed to reduce the dimensionality of very big chunks of data to visualize the data, like what I did here with the heat map. And then I have a function here in the utility functions that computes the t-SNE representation of a given matrix. I'll show here. Basically, this function takes a matrix where you have samples, in this case, samples on the rows and features on the columns. And then you compute it, reduce dimensionality, and then I just do some further decoration of the table that I get, and then change the column names to more, add some column names, and then return a data frame. By the way, again, I have to assume that you have some understanding of R and some background, otherwise I can't do any meaningful analysis here. That's why if you don't have any background, try to focus on the plots that I make, and it might be more informative. And later when you learn R, you can redo those things. And let's say if you run this, what do we get? If you compute t-SNE given gene expression, just gene expression data here. I'm just trying to explore the data here. This is what I get. It's basically a data frame with 300 rows and 2 columns. And 300 rows, okay, because I have 300 samples, and 2 rows, 2 columns with 2 reduced dimensions of this data. Basically, we reduce the dimensionality of the data from 1,000 features to 2 features. And let's say plot this with ggplot 2 and the ggplot, and then I provide a data frame, and I want the x-axis to be the first dimension, y-axis to be the second dimension. It will be a scatter plot, so I use geom-point and do this. Okay, now I have some plot where each point represents one patient based on only gene expression data. And then what else can I do to make more sense of this? Okay, maybe I can say there are some groups here, maybe they are more similar to each other. I can also overlay some additional information that I know from the clinical data, that it is call data. I have, what else did I have? Like a project, a subtype, I don't know, different kinds of information. So I could take that information and then map it to this table. So I have this table, and I know the row names of this table, which are the sample names. I can check which, and actually in this call data, they have in the same order, but I want to be able to make sure, I just want to make sure that I pick the right information for the right patient. So I match the row names of the table with the samples column in this call data object. And this is, by the way, a data table object. Data table, and then give me the project information. That's a project, in this case, it's the type of disease. And I have this information. Now I have a different table with the t-SNE information plus the type of disease. And maybe I could just plot this, right? And now I have some colors for each patient. Now, okay, it makes more sense. You have this esophageal cancer, maybe there's some stomach cancers. Then you see that maybe colon cancer and rectal cancers are more overlapping with each other than others. And then maybe you could also get subtype information with the same manner. Let's say how it looks like. And then there are some subtypes that come from a paper that analyzed the gastrointestinal cancers. And then you have these different subtypes. There seems to be a correlation of subtypes with the types of cancers. But yeah, these are grouped as a gastrointestinal cancers, and then anybody can come up with some subtypes, of course. You can come up with as many subtypes as you want, but somehow they have to be somehow coherent, somehow have some kind of commonalities in terms of their biological background. And then I'd like to show this nice package where you can combine multiple plots so that you can look at them next to each other. Let's say I run this plot, P1, and the P2 is for my subtypes. P1 was about the cancer types, P2 was about subtypes. and I can arrange them into the same place like this. Maybe let's change this type as a cancer types, and then maybe change this one as labs title equals subtypes, and then run it again, and then show it, and then yeah, basically yeah, you have a plot that you can maybe do a comparison of subtypes and cancer types in this space. In this case, it's about gene expression. And let's change this to GX again. I would like to compare this again to other layers. So, and then, okay, I want to do the same thing with just copy number variations and compute the t-SNE, add the project information, and then plot, okay? And then use this call plot function, plot grid function to plot them together. What I'd like you to see is that they are not always matching, right? The gene expression clusters might be slightly different than copy number variation. But of course, gene expression and copy number variation might be correlated more than gene expression and mutation. So there might be more correspondence. And then do the same with mutation data. Do the same with methylation data. And maybe use 4 of them, like gene expression, mutation, copy number variation, methylation. And then add some labels. And then yeah, basically you just see that different layers capture different kinds of information. Although like this group, like this esophageal cancer seems to be easy to distinguish for almost all data types, but maybe not so much for mutation data. And it seems that methylation data is the best predictor of different cancer types, maybe because, again, methylation data is very more fundamental in regulating which tissue types express which kind of cells. So methylation data might be more robust in terms of catching signals about, in this case, we are looking at tissue types, basically like colon and rectum and esophagus and the stomach. So it might be easier to cluster samples using just methylation data. So this is one other point that I'd like to make, is that you don't always, I mean, multi-omics doesn't always give you the best results, or integrating them might not always be the best. It depends on the objective that you are looking for. Maybe if you're just interested in tissue subtyping, maybe methylation is the best, or maybe for drug response, maybe you need some kind of specific mutation profile so that the drug works for that kind of disease. So, but sometimes maybe mutation is not good enough and you need gene expression on top of it. So, but without doing multi-omics, you can't know, of course, which one is best for which type of purposes. And then, okay, I did some data exploration, looked into the data. Now I'd like to do some multi-omics integration, this we talked about. And I will use this MCIA function. It's called multi-omics multi-co-inertia analysis, multiple co-inertia analysis. It's an extension of co-inertia analysis, which is an extension of principal component analysis. I use this library because it's quite simple to run and easy to install, easy to demonstrate. All the things that I show will be, I mean, more accessible to more people. And I think it gives reasonable results. You could also do way more advanced deep learning and stuff, but still, yeah, I mean, for beginning, maybe this is good enough. This gives you good enough results for a lot of the things that to start with. So this is how I integrate. So basically, all you need is a list of tables, omics tables. Here I'm subsetting these tables that I had for mutation, copy number variation, gene expression, methylation. And you need to tell how many, what kind of reduction you want to have. Like in the end, how many meta features or factors you want to get in the end. Now it will be more obvious to you what I mean. Like here, this command integrates the layers and then gives you some meta features, which I'll call latent factors. This should run in about like 30, like maybe 15 seconds or so, depending on the environment you have. Compute power, it might be a bit different. Okay, it's done. So it returns this integration function object, this MCIA class. It has some information in it where I'm interested in this one, basically. This latent factors here. I'll show you how it looks. Latent factors. And basically, it's, as I said, when I said 10, it gave me 10 factors. In the end, I have a column, a table with 10 columns. If you look at this latent factors, I have 300 samples and 10 dimensions in this data. Before I had how many? I had like 1,000 each, which would be like 4,000 features for 300 samples. Now I have 300 samples and 10 features. And I just do this for sometimes in R, the namings of columns are like these sample names normally have these dashes, but they were turned into dots because they are not allowed in some of the algorithms. So here I'm changing the dots back to dashes again later, because because the names of the samples I have has these dashes. Then later when I want to match them, they won't match if I keep them as it is. So I just replace them. And if I look at the matrix, okay, now I have these dashes in the namings of the samples. And again, I want to just visualize this t-SNE, maybe with the integrated data. Compute the t-SNE. Let's see how it looks. Again, I have this 300 samples and 2 dimensions. Then I want to visualize the type of cancer with these dimensions. And then this is how it would look with the latent factors integrated, using only 10 features. But this is now like a mixture of 4 different omics layers. It looks similar to other layers. Again, it's mostly dominated by methylation information and some gene expression. Again, you see esophageal cancers a bit different then, and like rectal cancers look clustered more with the colon cancers. And what else you can do with this latent factors? You can do some downstream analysis, like you can do if you have some information about, let's say, each patient has a drug response value, you can try to predict those drug responses. Or before going into that prediction, maybe you can just visualize those things. Like, let's say, I want to get the age distribution of the samples here. This is what I do. And maybe I want to check if some of the factors that I come up with here actually correlate with age, for example. I don't know why you want to do that, but maybe you would like to see there some of the factors might be explaining aging, like there's age-specific differences. And one thing you can do, again, you can do a correlation between a simple, let's say this is how you would do it, latent factors. And then maybe take one of those and then look at the correlation with age. But I need to, some of them may have missing observations. That's why you use this only the samples with the information about age. And then, so this is how you do it for one column, but I want to do a loop. I want to do it for each column, like the apply. I use this apply function, do this. 2 means do it on the column wise, and then instead of row wise, and then basically go through each column of this table, go through each of them for each factor, basically, and look at the correlation of each factor with the age of each, with the age of the samples. And then this will return this is a return basically correlation scores between each factor with the age of the samples. And then, but I'd like to sort I'd like to find which ones have the most correlation. Maybe, and I'm interested in positive or negative correlation. So that's why I take the absolute values of these. Then I want to sort them by decreasing order. And then you see, okay, you see a sorted list of variables that are most correlated to aging, to age factor. And you can also visualize this. Let's say just take one of them, and you can make a data frame like this, where you have the variable that you found that there's the most correlation and the age. Then I use this ggScatter function from this library because it has some additional things that I can use. I can do this like this. Sorry, and I need to tell what is the x factor, what is the x-axis, and what is the y-axis. And I know this is the var1, and then gives me this kind of scatter plot. But I'd like to also add some regression line, basically what is the trend with this plot. You can use this. And I'd like to also plot the correlation coefficient here. What happened? Okay, yeah. So you see this, now you have a scatter plot of age versus the latent factor that you found, the most correlated. So there's a negative correlation with this factor with aging. So, I mean, this is a simple thing that you can do individually for each factor. But you can also, what you can also do, you can build more machine learning models where you can combine multiple factors to predict age profiles, for example. You can also do something similar with tumor stage. I'm not going to go into too much detail, but in this case, age was a continuous variable with numbers, but tumor stage is a categorical variable, right? If you look at this, it's a categorical variable. But I still can do some kind of statistics to figure out, okay, which factors are most enriched are most different across different tumor stages. And let's say I make a table here and adding information about, okay, I have my factors here, latent factors I found, and I have a stage information. And I'd like to change the shape of this table by melting. It's called melting. You basically take first factor versus stage, second factor versus stage, third factor versus stage. You can do that by basically with this melting function. What you get is that, and I'm focusing on only samples that have a stage information. And this is the kind of table that I have now. Yeah, you have the factor. Basically, I concatenated each factor on top of each other. Then I just want to plot this. Again, ggpower has this nice box plot function where I can plot a categorical variable with a continuous variable, right? Because my latent factors have continuous values, but the tumor stage is a categorical variable. So a nice way of visualizing it is a box plot. So you can do, if you do it like this, and let's say this is how the bare minimum would look like. You would have a box plot, but it doesn't help a lot with just looking at these box plots. Maybe add some information about like some coloring of the stages, maybe add some, but again, here we are looking at all factors versus all age, so we'll need to separate by the factor. So I want to have one box plot per factor versus age. That's what I do here, faceting a table plot. And I also want to add points, like the actual observations, into the plot. And this is how I get. So now I have a box plot separated by each factor with points, and then with stages on the x-axis and values of these different factors on the y-axis. And I can also regulate how many rows I want to have here. I could have, okay, 4 rows, for example. It will make me 4 rows. Or let's say only 3 rows, and then it will squash different things. And then also I'd like to, let's say, stick to 3 rows and then do the statistical test for each group. So this is a out-of-the-box easy exploration methods. Basically, for each factor, I'm checking if there is a distributional differences between age stage groups, and then what is the p-value for this. It's basically like one-way ANOVA, doing categorical, like checking for differences in distribution for a continuous variable versus a categorical variable. And then maybe the rest, I find there is 2 variables, like this one and this one, associated to differences between tumor stages, maybe I plot one of those, let's say the fourth one, to see the difference a bit easier. You see that maybe that factor is capturing some differences. Maybe it could be useful in figuring out which samples are differences in terms of that arise because of tumor stages. Might be separable by this kind of factor. And tumor stage, if you have tumor stage 3, maybe it's higher, and stage 0, stage 1, that's lower values. So these are kind of sequential exploration of your latent factors. That you can also do whatever. You can also do this with gene expression and other kinds of features that you have. But here we have few things to do. So I mean, like, we reduce the dimensionality, so it's easier to do. I guess I'm running out of time, so maybe I'll quickly show what I do here. Okay, because for questions we are running out of time. So one other thing I said we could do is clustering, and here I do some. If I plot the integrated latent factors, I color them. You can use this package for exploring what is the optimal number of clusters and measure them by certain evaluation criteria. And you can use this library for this experiment. And then you can find the optimal number of clusters using different criteria, and then find, okay, what is the best cluster for certain criteria. Then I pick, okay, let's pick the clusters as 5 clusters looks nice for maybe multiple clustering measures. And then I add the clustering information, and now I can see the kind of subtypes that I found myself by clustering my samples on my own, with my own data. And then, okay, I'm not going into this detail about, okay, there's a function where you can find out, okay, I found these clusters, but what is different between these different clusters? You can run them. Maybe I quickly run them, and then I find, okay, this subtype is associated to this variable, maybe, and then I use those to plot them. Then basically, I found this first variable associated to certain clusters number 1 and 5, and then I could see that this variable is enriched in these 2 clusters. I can visualize it. Okay, I'll skip this other clustering method, that's integrative clustering. There is some ways of evaluating how to evaluate which classes are better based on known criteria and visualizing them. And some survival analysis. Maybe I put too much too much material here, but I wanted to have them in place so that you can later work on them, work through the code and make sense of it. We can also make sense of this. We can also use this survival data here to distinguish between different clusters, and then see if they make sense in terms of survival differences. For example, here I'm building a survival model, and then using the clustering information, I see that some clusters have less survival compared to other clusters. Maybe cluster number 1 here, fewer people, more people make it past 2,000 days after diagnosis of a cancer, but with cluster 5, the maybe average survival time is about 500 days for most of them. And you could do the same for other clustering methods and then compare them. Okay, which ones gives you the best clustering? So, okay, you can also do classification. Maybe this is important. Okay, I will show this one, like how you can train a random forest and for classification or regression. And you can use the same code for classification or regression tasks. Here, I'll just show one classification task. And here is the function, train random forest. What we are doing here is that given a matrix and some outcome variable like labels, I split the matrix samples into training and testing samples, and then run this library called Ranger for training a random forest on the training data, and return model that learns from the training data and also return the data that it's split for as testing. So the testing data is not seen by the training algorithm. And here I'd like to use the subtype information as the labels of samples. I'd like to use the latent factors that I got from this integration, and then run this training algorithm on this, train a random forest on it. And then what I get is basically as a fit. This is the model you get that you can use to test on this test data. And the test data is basically the 30% of the actual data that we started with. And then you can use this function basically that has the model and the new data. And then you can do a prediction on the new data. And then the prediction object will contain your predictions for each sample in the test data. And then you can compare what you predicted with what you knew from the test data. I mean, you already knew that labels, but you didn't know how well your model worked on this data. Then you can make the table. You can see, okay, when for samples that belong to this subtype, I accurately classified most of them, but some of them are wrong. So you can basically, you need to evaluate how well you perform on this test data using different accuracy measures, using different evaluation metrics, which you can use again here, the Caret library to do a confusion matrix evaluation. And then you can get these specific metrics for all sorts of like specificity, sensitivity. Like here we can say that we have an 80% accuracy, and if you just randomly guessed, we would have an accuracy of 0.27. So it's much better than random guess. That's what you can say. So here I show an example of regression for predicting the age. I'll skip that. And the second major exercise was about drug response prediction. Maybe I'll quickly show again. This is data from CCLE. Again, I check. I have data for mutations, copy number variations, gene expression for 658 cell lines and with differing number of features and some drug response information. And this is how the drug response information looks. Again, this is a problem of regression because it's a numerical variable. And I would like to see how the drug response is distributed. Now, I could just use the same idea that I used before, training random forest, but in this case, it's a regression problem. By the way, this is a utility for this ranger. It doesn't work the same way for other packages you would use. Here, I'm taking the drug response values as the outcome variable. You see this, there is 600 something measurements. And then I'd like to do, what I demonstrate here is that you could integrate data for drug response prediction and using different layers of omics data. In this case, I integrate using mutations and copy number variations, and then call it as a genomic features. And then I could also use genomic features plus transcriptomic features, and then do an integration, and then use that to predict drug response. And then see that maybe if you, what I'm trying to show here is that if you add this additional layer of transcriptomic data for this drug, you can predict the drug response much better than if you just use genomic information where you use mutation data and copy number variation data. So these things I will run quickly and then maybe show you. Okay, I'll just leave it there probably for questions. I'm sorry that it was too much material maybe, but hopefully you can go through this material with this quick introduction. Of course, we have to assume that there is a lot of background. You would need some programming experience, some information about how sequencing works, how these measurements work, some information about how machine learning works. So hopefully these different applications, basically like clustering, classification of subtypes, survival modeling, and drug response prediction. So all this material is here. So hopefully you can go through this material and then learn from it. So here I ran this two different integrations, like one it is genomic information, one with the genomic information plus gene expression, and I like to train again. I'm running it again, but and I'd like to compare the known drug responses with predicted drug responses, and then plot them here. You see this, if I just use mutation and copy number variation data, I get a correlation of 0.17, which is okay. But if I train a random forest on mutations, copy number variations plus gene expression, then I get a much better correlation that you could get to predict this drug response. So maybe I just leave it at that. Here, what I do is that this process could be repeated many times, and then you could have this, because every time you do a train test split, you will get slightly different results. To have an understanding of how generalizable this is, maybe you could repeat the whole analysis multiple times, and then do this comparisons, which should show that on average for any kind of splits of training and testing, that you get better results with if you have additional layer of information for this specific drug. Okay, I'm sorry, maybe I took too much time. Maybe we get to questions, maybe get a couple of questions. If there is some demand, maybe I could also later I could do something like this with in Turkish if there is some demand, or you can ask me more questions later as well.
Çok teşekkür ederiz hocam. Dilerseniz YouTube katılımcılarımızdan gelen soruları size yöneltebilirim.
Evet.
İlk sorumuz, İngilizce soracağım hocam. What is the risk of making genetic configurations to prevent these cancer types?
Can you repeat it again? Sorry.
What is the risk of making genetic configurations to prevent these cancer types?
Risk of genetic configurations. I don't understand the question properly. What is the risk of...
İsterseniz soruyu soran arkadaşımız bunu daha açık bir şekilde yazabilirse daha sonra döneriz.
İsterseniz Türkçe'ye de geçebiliriz eğer sıkıntı oluyorsa olabilir. Zaten hani...
karışık sorularımız var hocam.
Öyle mi? Tamam. Aslında belki ekrana baksam ben de. Şimdi burayı kapatsam nasıl olacak? Bir saniye.
Tabii ekranda da görebilirsiniz soruları.
Tamam. Bazı mutasyonlar kanser yol açıyor. Evet. Yani mutasyonlar, yani aynı mutasyonlara sahip olsanız bile bazı insanlar kanser geliştirmiyor. Bazıları geliştirebiliyor. Niye? İşte tumor microenvironment dediğimiz yerde sadece kanserli hücreler yok. Başka immün savunma hücreleri var. Savunma hücreleri kanserli hücreleri tespit ediyorlar. Savunma hücrelerimiz de birbirimizden farklı. Herkesin savunması birbirinden farklı olabilir. Veya işte dediğim gibi çok heterojen bir ortam olduğu için bazı kanser hücreleri sadece mutasyonlar değil, daha başka modifikasyonlar geliştirmiş olabilir. Bu immün sistemden kaçma şansı olabilir. Başka işte mikrobiyom var. Yine dediğim gibi mikroplar var bu tumor microenvironment içerisinde. Onların aktiviteleri bu şeyde etkili olabilir. İşte herhangi bir ilacın işte nasıl proses edildiği, nasıl emildiği, nasıl işte oraya gittiği bu ortamla da ilişkili olabilir. Yine oradaki kompozisyonla ilişkili olabilir. Tabii bir sürü faktör var. Yani o yüzden demek istediğimiz yani sadece mutasyona uğraman yetmiyor yani kanserin oluşması için. Daha başka bir sürü faktör de bir araya gelebiliyor. O yüzden sadece mutasyonlara bakmak kanseri anlamak için yetmeyebiliyor. O yüzden.
Cevabınız için teşekkür ederiz hocam. Bir sonraki sorumuz ekip arkadaşlarımızdan geliyor. Makine öğrenmesinin metabolomik verilerinin diğer omik verileriyle bütünleştirilmesi neden zor olmaktadır?
Zor değil. Yani muhtemelen zor değil. Aslında benim metabolomik data da pek tecrübem yok ama özünde yine belli metabolitler var. İşte onların belli ölçümleri var. Metabolitleri işte tespit belki ölçmenin mass spektrometri falan kullanıyor yanılmıyorsam. Proteomics'te de öyle. Belki daha az metabolit bulunabiliyordur. Ama sonuç olarak özünde belli numerik ölçümleriniz var. Farklı metabolitler ve farklı sample'lar üzerinde. Bunların dağılımları farklı olabilir. Tabii ki zaten bütün omics katmanlarında farklı dağılımlar var. Zaten bu integrasyon araçlarının özelliği de o. Yani onları aynı şeye indirgemeye çalışmak, aynı ortama indirgemeye çalışmak yapılabilir. Yani zor değil. Sadece onunla ilgili data olmayabilir. Yeterince data olmayabilir. Bu TCG, benim gösterdiğim TCG ve CCL'de en çok işte kullanılan şeyler orada onlara bakılmış. Yani metabolik data çok fazla yok. Olursa daha iyi tabii ki yani. Olursa daha iyi olur tabii ki. Kanser çünkü metabolizmayı kullanıyor.
Teşekkür ederiz hocam. Bir sonraki sorumuz YouTube katılımcımızdan geliyor. Chemotherapy or other treatment methods cannot overcome cancer stem cells. That's why recurrence of cancer is observed. Is there a way to destroy cancer stem cells?
Yani şey değilim, tam olarak bilmiyorum açıkçası ama hani muhtemelen özünde yine şeydir. Belki bu işte yeni mRNA teknolojileri kullanılabilir daha spesifik atış yapmak için. Çünkü sıkıntı evet dediğiniz gibi kanser kök hücreleri var dokuda. Gerektiği anda istediği kadar çoğaltabiliyor, kendisini kopyalayabiliyor. Ve hızlıca çoğalabiliyor. Bir de kök hücre yani dedifferentiation deniyor. Yani normal dokudaki işte kas hücresi kök hücreye dönüşüyor. Daha sonra sonsuza kadar çoğalabiliyor. Yani onları yok etmenin yolu belki işte dediğim gibi immün sistemi ona karşı tekrar eğitmekle ilgili özünde mümkün olabilir. İşte bu CAR-T terapiler var işte hastadan işte immün hücreleri alıyorlar. İşte onu laboratuvarda genome editing işte CRISPR gibi yöntemlerle editleyip işte çünkü hastadan aynı zamanda kanserli hücrelerini de tanımak gerekiyor. Bu kanserli hücrelere göre eğitip daha sonra tekrar hastanın vücuduna enjekte ediyorlar. Bunun ve o kanserli hücrelerin saldırmasını sağlıyorlar. Belki oradan bir avantajı olabilir. Ama dediğim gibi yani bu konuda çok detaylı bilgim yok. Hani özellikle kök hücreleri nasıl tespit edilebilir, nasıl saldırılabilir? Muhtemelen onunla ilgili çalışmalar vardır.
Teşekkür ederiz hocam cevabınız için. Ekip arkadaşlarımızın hazırladığı sorularla devam ediyoruz. Genler ve omik katmanlar arasındaki regülasyonun dinamik modellemesi, makine öğrenimi tabanlı çoklu omik veri entegrasyon araçlarının gelecekteki gelişimi için umut verici olabilir mi?
Evet, yani aslında bütün bu yani sunumun biraz da o yani temel motivasyonu. Bu bahsettiğim işte Deep Medicine denen kitapta da mesela onlar anlatılıyor. İşte hani sadece omik veriler değil, diğer başka katmanlardaki verilere de erişebileceğiz bir süre sonra. İşte giydiğiniz işte kolunuza taktığınız Apple Watch'lardan ne bileyim işte bir sürü değişik wearable araçlar olacak. İşte giyilebilir araçlar, işte çevreyle olan temasınız, işte iklim, moleküler veriler, multi-omics veriler bunların hepsi bir araya gelerek tabii entegre halde. Bu tabii hatta daha da şey olması lazım. Bir sonraki aşama tek hücre düzeyinde anlamak. Tek hücre düzeyinde anlamak da yetmez. İşte bu 2D special işte bir sonraki workshop'ta göreceksiniz single cell analiziste. Bunun işte iki boyutlu 2D iki boyutlu analizi de var. İşte diyelim patolojik slide'dan bakıp bunun bir sonraki aşaması 3 boyutlu kanser modellerini anlamak. Yani evet gelecekte öyle olması lazım. Yani bu datanın üretilmesinin kolay, ucuz, hızlı olması lazım. Yani bu kanseri tam anlamak için yani. Çünkü dediğim gibi bütün katmanlar farklı veriler üretiyor. Herkes de farklı nedenlerle ortaya çıkabiliyor. Yani en iyi anlamak için bu daha yakından bakmamız gerekiyor.
Cevabınız için teşekkürler hocam. Bir sonraki sorumuz YouTube katılımcımızdan geliyor. About half of the genome consists of transposon repeats and they can alter gene expression, loss of function or gain of function. In cancer omics, is it possible to analyze the molecules?
Yani burada aslında daha iyi açıklamak isterdim Copenhagen aberration, somatic Copenhagen aberrations denen konu aslında biraz bunu ölçüyor. Yani orada genomda duplication'lar var, deletion'lar yani amplifikasyonlar veya bazı bölgeler siliniyor, yok oluyor. Bazı genlerin kopya sayısı azalıyor veya işte bu repeat'lerin bu microsatellite instability denen var. Orada da yine bazı yani bütün genoma bakmadan bazı repeat bölgelere bakıp hani bu genomda ne kadar bir olmuş. Bu tarz aberration'lar ne derler? Genomdaki bozulmalar. İşte bu yüzden İngilizce sunumu İngilizce yapmamın sebebi biraz da oydu. Türkçe'ye zor.
Dilerseniz İngilizce'de de cevap verebilirsiniz.
Yok yani sorun değil. Artık karıştı. Evet yani amaç bunu da indirekt olarak ölçen, muhtemelen direkt olarak ölçen şeyler de vardır. Omik yöntemler de vardır transpozonlar için. Ama dediğim gibi bu dataların hepsi şey olmuyor. Halihazırda bulunmuyor. Data bulmak zor. En büyük bahsettiğim konsorsiyum, yani halka açık olan datasetlerde en çok kullanılan, en yaygın, en bilindik datalar var. Ama bu bahsettiğiniz repeat gibi konuları çalışmak için de biraz işte copy number variation, microsatellite instability gibi şeylere bakarak indirect yoldan sanıyorum bir şey yapılabilir. Analiz yapılabilir, evet.
Cevabınız için çok teşekkürler hocam. Tekrar YouTube katılımcılarımızdan gelen sorularla devam ediyoruz. Merhaba hocam, öncelikle sunumunuz için teşekkürler. Farklı omik verilerin farklı dağılımları ve boyutsallıkları varken verilerin entegrasyonunda nasıl bir yaklaşım uygulanmaktadır?
Yani evet, bu şey tabii bahsettiğim linkini, yani bahsettiğim review makalelerine bakarsanız benchmarklar var. Bu bir sürü değişik omik yöntemler. Bahsettiğim işte matrix decomposition yapanlar var. İşte bir değişik omics layer'ları matrix decomposition'la aynı zamanda dimension reduction yapar. Bu lineerce bir gerektiren bir yöntem. İşte deep learning metodları var. İşte bu hani bahsettiğim autoencoder'lar. Yine orada da bir sürü değişik assay'i üst üste ekleyip kompres ediyorsunuz datayı. Orada yine datanın esansını elde tutmaya çalışıyorsunuz. Yani bu soruya tabii kısa bir cevap vermek çok zor. Bunun için şeye girmek lazım. Makaleleri ayrı ayrı okuyup her bir metot nasıl yapıyor? Hepsinin matematiksel olarak farklı yöntemleri var. O alanda yani uzmanlaşılabilir tabii ki. Yani bir sürü işte deep learning, linear algebra, yani temel klasik machine learning yöntemleri. Klasik işte principal component analiz de bunun bir şeyidir. Farklı boyutsallıklar, yani bu multi-omics integration araçlarının üretime nedeni biraz da işte bu bahsettiğim farklı boyutsallıklar ve farklı dağılımlar. Mesela işte bu canonical co-inertia analizisi yapan, bu örneğini verdiğim, R'da gösterdiğim pakette şey yapıyor. Her bir omics katmanını principal component analiziyle benzer bir şeye indiriyor. Benzer dağılım olmasını sağlıyor. Ondan sonra entegre ediyor. Yani bu farklı algoritmalarda farklı olabilir. Bir sürü değişik yöntem kullanıyor olabilirler. Biraz uzun cevap da vermiş olayım aslında. Kısa cevap olan bir soruya.
Teşekkür ederiz hocam cevabınız için. Bir sonraki sorumuz. How can the reproducibility of latent factors derived in deep learning methods be ensured?
Evet, bu doğru. Deep learning yöntemlerinde şey oluyor. Bu diğer bahsettiğim işte matrix factorization yöntemlerindeki gibi tekrarlanabilir, reproducible olmayabiliyor. Birebir mi reproducible olmayabiliyor. Bunun için işte yöntemler olabiliyor. İşte değişik regularization teknikler deniyor. İşte yani deep learning algoritmasına verdiğiniz inputların işte mümkün olduğunca belli bir en önemli kısımlarına fokus olmasını sağlamaya çalışabiliyorsunuz. İşte veya mesela aynı yöntemi tekrar aynı data üzerinden birden fazla kez çalıştırıp ortalamasını alabiliyorsunuz. O da mesela reproducibility, yani beklenen umduğunuz sonucu ortalama olarak verme olasılığını artırıyor. Doğru, bu yani deep learning metodlarında bir diğer klasik matrix factorization yöntemlerine göre bir dezavantaj sayılabilir. Ama deep learning metodlarında da daha non-linear ilişkileri yakalama şansınız olabiliyor. Bu matrix factorization yöntemleri daha lineer ilişkileri tespit etmenizi sağlıyor.
Cevabınız için teşekkür ederiz hocam. Bir sonraki sorumuz ekip arkadaşlarımızdan geliyor. Hasta sınıflandırılmasında mevcut kümeleme algoritmalarının ne gibi kısıtlamaları bulunmaktadır?
Evet, bu herhalde clustering ile ilgili. Hani evet, sınıflandırıyorsunuz. Sınıflandırmanın veya kanser subtyping denen şeyin bir dezavantajı, dediğim gibi hangi omics layer'ı kullanırsanız farklı bir sınıf çıkabiliyor. Veya hangi ilaç içinse, yani ilaç treatment A için farklı bir sınıflandırma çıkabilir, treatment B için farklı bir sınıflandırma çıkabilir. Yani farklı bir grup, farklı A grubu A ilacına tepki verirken B grubu işte B ilacına tepki verebilir. Ama bu A grubundaki ve B grubundaki hastalar diyelim mesela survival açısından işte yine benzer ortak noktalar olabilir. Yani konu şeye dönüyor. Yani amacınız ne? Yani neden sınıflandırıyorsunuz? hangi faktörlere göre sınıflandırıyorsun? Yani hangi omik datayı kullanarak sınıflandırıyorsun? Hepsinde farklı sonuçlar alacaksınız. Yani büyük ihtimalle olabildiğince farklı sonuçlar alacaksınız. Farklı layer'lardan. O yüzden işte bu Integrative Clustering denen yöntemler bunların bir konsolidasyonunu sağlıyor. Bir o amacı var. O fonksiyonu var yani. En azından hani genel olarak bütün bu farklı layer'lara baktığım zaman en ortak, en esansı, bu hastalığın esansını daha iyi kavrayabilir miyiz diye. Ama evet, kısıtlaması bu. Bir de en iyi şey nedir? Clustering. Yani 3'e mi bölmeliyim? 4'e mi? 5'e mi? 10'a mı? Kaç tane subtype var? Bunların işte bir sürü evaluation metrikleri var. Şeyde de gösterdim. Kodlarda da var. Ama orada da göreceksiniz. Bir sürü değişik metrikler, değişik cluster sayısı önerebiliyor. Birisi 5 tane diyor, birisi 3 tane diyor. Yani günün sonunda herhalde sadece clustering yaptım. Tamam, bu budur. İşte 3 tane subtype var demek tek başına anlamsız. Neye göre 3 tane subtype var? Yani belli bir ilaca göre tepkiye mi? Survival'a göre mi? Ya işte bu transkriptoma göre mi? Epigenoma göre mi? Integrative mi? Yani bunların hepsine bakmak lazım.
Cevabınız için çok teşekkürler hocam. Şimdi son sorumuzla kapatıyoruz artık. Hedeflenen tedavilerden en çok kimlerin faydalanacağını belirlemek için hastaları farklı moleküler imzalara dayalı olarak sınıflandırmada sistem biyolojisi yaklaşımı nasıl bir fayda sağlamaktadır?
Yani evet, sistem biyolojisinin amacı da bu zaten. Yani, şey yapmayalım. Yani tek bir gen çalışmayalım ömür boyunca. Mümkün olduğunca bu işin bir hani kanser kompleks bir hastalık. Sadece moleküler şeylere bakmak da yetmiyor hatta. Hücre biyolojisine bakmak, hücreler nasıl etkileşiyor onlara bakmak lazım. Organ sistemleri nasıl çalışıyor, birbirleriyle ilişkisi nedir? İşte dış çevreye olan ilişkisi nedir? İnsanın işte yediği yemeğin etkisi nedir? Yani diyetin etkisini, yani bütün bu faktörleri bir araya getirdiğiniz zaman belki de en anlamlı sonuç çıkarabileceksiniz. İşte sistem biyolojisinin amacı da zaten bunu sistematik olarak bütün bu faktörleri bir araya getirerek anlamayı sağlayacak araçları bir araya getirmeye çalışıyor. Yani o yüzden hani tek işte klasik yöntemde işte nedir? Bu özellikle kalıtsal hastalıklarda işte sickle cell anemia, okey, bir tane hemoglobinde bir tane mutasyon var, tamam onu çalışayım gibi olmuyor yani şeydeki. Yani küçümsemek için söylemiyorum, sadece şeyi farklı. Yani kanserin kompleksliğini anlatmaya çalışıyorum şey olarak, moleküler bazda.
Çok teşekkür ederiz hocam. Değerli vaktinizi ayırdığınız, bilgilerinizi bize aktardığınız ve bilgi dolu bu eğitim için kendim ve tüm katılımcılarımız adına çok teşekkür ederim.
Ben
Sizlere
de çok
sağlıklı günler diliyorum. Sağ olun
ben
hocam.
çok teşekkür ederim. Davetiniz için de kusura bakmayın, biraz zaman fazla harcadım. Biraz fazla materyal vermeye çalıştım ama umarım faydalı olur. Herkes kendine fayda olacak bir şeyler bulur
Çok sağ olun
materyalden.
hocam.
Görüşmek üzere.
Teşekkürler. Sağlıklı günler.
İyi günler.